Good morning and welcome to StudentSOS, or welcome back if you've been here before. Thank you very much for joining us either way. I think I should explain the picture again in case you haven't seen this one before. I hate having selfies taken and I despise being on video, which makes me a very odd choice as a YouTuber. However, a very good friend of mine sent me this card and it is a selfie and it seemed exactly appropriate for these types of content. So instead of seeing my face at the start of these videos, at least for as long as my editor tolerates it, then we're going to be able to have a look at the selfie and we'll try and update with some newer pictures. So this is StudentSOS. It's very nice of you to join us this morning. If you haven't already, please click the like button and subscribe so that you can be updated with new material as it's released. We are releasing 2 videos a week currently, and that will hopefully be on a scheduled day. I will give you details of that as and when. But we have a new channel looking to help students from the 11 plus all the way through to BMAT and UCAT examinations. We are science focused after the 11 plus at Key Stage 3 and GCSE across all exam boards, and at A-level we are currently working on A-level biology. The material that I've been recently uploading is AS level, so year 12 biology, first year A-level biology. This will be useful to year 12s headed into year 13 in September, or even for you year 11s who are very keen to study this subject next year. It does no harm at all to get a handle on some of the new language that you're going to have to acquire during the 2-year course for A-level biology. Fun fact, apparently A-level biology is a subject with the most new words in it across all A-levels, including, by the way, all modern foreign languages. So let me just say that again. The subject that has the most new language in it at A-level over a 2-year course is A-level biology. That includes all A-level language courses. I was told that by a biology examiner many years ago when I started examining. So fairly astonishing amount of material that you've got to get a handle upon. It's never too early to start. So without further ado, we're going to do the last of the biological molecules videos today. We've taken a look through carbohydrates, so the monosaccharides, disaccharides and polysaccharides. We've taken a look at lipids, and today we're going to take a look at proteins. We've already done a separate video on DNA structure, and we will do a separate video on biological food tests within the practical series of videos. I am aware that lots of you will be struggling with the practical aspects with everything that's been going on in the world over the last 18 months. So that is something that is a priority on my list over the summer, to address the practical aspects of the A-level biology course in all specifications. So this morning, we're going to take a little look at proteins. So we have the basic structure over here of an amino acid. So the monomer of a protein is an amino acid, and there it is. You need to be able to draw and label this structure. So we have your amine group, your NH2 group on the left-hand side. We have the carboxyl group on the right-hand side, and we have the R group. This is my variable group, and this will vary depending on the amino acid I'm attempting to code for. Quick note here, you will not need to learn any of the variable groups and the amino acid they are associated with. You need to be able to draw the basic structure of an amino acid and know how to label one as it's drawn here. You do not need names of amino acids. They will tell you them in the exam. And you do not need to know the variable groups each amino acid has. There are only 20 amino acids that are used to make proteins. That's all. And we took a look at how this works in terms of DNA base sequence, A, C, T and G, in the DNA videos I've already made, so please go and take a look at those. Something you might need to do on an exam paper is how we form a peptide bond. So when we join amino acids to produce dipeptides, we need to form a peptide bond. If you've already watched the carbohydrates video, you'll have a good idea as to how this works. What we do in the exact same way as carbohydrates is we remove one molecule of water in a condensation reaction, and that produces a peptide bond between the C and the N, the carbon and the nitrogen. It's a peptide bond formed by condensation, broken by hydrolysis. So we add a molecule of water back in and we separate the dipeptide into the 2 amino acids again. So you could be asked in an exam to be able to draw exactly as I've got here, the removal of water, the formation of a peptide bond, and to name the peptide bond. Couple of marks, make sure that you can do it. Very straightforward. So one of the topics that seems to cause quite a lot of distress for students within proteins isn't so much amino acids and the peptide bond formation, but the structure. And I'm never really very clear why. So I'm going to take you through the structures now. They're not very complicated. I accept that they're possibly not the most fun that you can have in biology, but nevertheless they are important. And again, as I mentioned in the polysaccharides video, unfortunately, the less excited you get by these things, the more examiners seem to want to put them on your exam. Please do not swerve A-level biology content that you find dull. It'll almost certainly turn up on the exam that you sit. So a primary structure is really simple. This is what's produced out of the top of a ribosome. I have some videos coming shortly on transcription and translation, but it's just a chain of amino acids joined by peptide bonds. So in other words, lots of that in a chain. And the reason these guys are all different colours on the diagram is just that they represent different amino acids. But you could have a chain of the same amino acid. You could have 3 of one, 2 of another, 1 of a different one of another one. This all depends, of course, on the DNA base sequence. So there are many permutations of a primary structure of amino acids, but this is what you need to know. It has only peptide bonds. Know the bonds in each level of the structure. So then we move on to the secondary structure. And you have 2 main types. You have alpha helix, or the alpha helices, and the beta pleated sheets. I apologise for my diagrams immediately. Your textbook will almost certainly have better versions of these, but you know what I'm looking at here. So alpha helix, beta pleated sheet, but both of these guys contain hydrogen bonds within them to hold the structure together. And of course, because they're made of a primary structure, they also contain peptide bonds. They're the same peptide bonds that were in the primary structure. Now, proteins can be made up of either alpha helix or beta pleated sheet. Generally, beta pleated sheet are used more in structural proteins, and the alpha helix is used more in globular proteins, so ones like enzymes and hormones. However, you can also have, and many proteins are made up of some of both. So you may find alpha helix and beta pleated sheet in the structure of the same protein, and that's completely acceptable. So once again, the bonds you're interested in here at secondary level are hydrogen bonds and peptide bonds. And so we move on to a tertiary structure. This is our 3rd level of folding. So this is further folding of the secondary structure. Regardless of whether we do globular folding, so many proteins are folded this way, and many of them contain both. I've written in yellow above. Many proteins contain both, so just like many proteins are alpha helix and beta pleated sheet, many proteins contain globular sections and fibrous sections. Again, however, fibrous proteins tend to be more your structural proteins. I've given you an example there of collagen. And your globular proteins tend to be more your enzymes and those types of protein. Regardless of which type you have, either way, they will contain hydrogen bonds. They have those from the secondary structure, but they will also form additional hydrogen bonds between OH groups and H groups on neighbouring amino acids. Disulfide bonds, links, disulfide bridges, it's all the same thing between cysteine residues. Now, I've just told you, you do not need to know the names of any amino acids. And that's the truth, I wasn't lying. But cysteine residues contain sulfur. And so it doesn't hurt to know that if you've got 2 cysteine residues that end up lying close to one another in a tertiary structure, they will form disulfide links, bonds or bridges. Only neighbouring cysteine residues can do that. You'll also form ionic bonds. Hopefully you remember ionic bonds from GCSE chemistry, or if you're lucky enough to be an A-level chemist, you'll know lots about ionic bonds. But in other words, some of the amino acids have slight positive charges, others have negative charges, and if they find themselves relatively close together as we start to fold this protein, they will form ionic bonds between neighbouring variable groups, R groups, in different amino acids. And likewise, hydrophilic and hydrophobic interactions. Now, these are something that you don't really know a great deal about because you don't learn about the amino acids individually. What you probably don't know is some of them like to be away from water. They're hydrophobic. Now, these guys are going to be found on the internal surface of the protein. They're not going to be near the outside where the water is. Now, the hydrophilic amino acids are going to find themselves on the outside edge or in the active site if it's an enzyme because they love water. They want to be near water. And so the folding enables the amino acids with their properties in terms of their attraction or repulsion for water to be adhered to. And so these are due to interactions between different R groups on different amino acids. You get these hydrophilic and hydrophobic interactions and the ionic bonds and the disulfide links. But all of these can and will be contained within a tertiary structure. So again, it's the bonds you're really interested in here. And lastly, not all proteins form a quaternary structure. It's important to start with that. A quaternary structure occurs when any protein is made up of more than one polypeptide chain. And here we have hemoglobin. There are 4 subunits. I appreciate you'll never be able to replicate that diagram. Such is its accuracy. You can see little green blobs. The one on the bottom right-hand corner looks less green. But little green blobs, they are your heme groups, your iron groups. There's 1 per subunit, and they are 4 subunits. They were made up as 4 different tertiary structures that were joined together in a quaternary structure to produce hemoglobin for the carriage of oxygen around the body. It is not the only quaternary structure that exists, but it's certainly the one that you would need to know as an AS candidate for A-level biology. So if you have any questions on any of this material, please drop us a comment below. Click like if you enjoyed the video and subscribe to continue to follow our journey through this course. The stuff that I'm covering here covers all A-level different specifications. When there are variations in the specification, I will highlight them, and very occasionally I will make separate videos because the content level is different for one exam board compared to another. In the meantime, thank you very much for watching this morning. I hope you enjoyed the video. Have a great day.